Plant Molecular Genetic Workshop. 2 classes, SBI4U, went to McMaster University on May 16th, 2023. We were met by 2 profs at the Faculty of Science to work on genotyping and gene expression analysis. The aim of the workshop is to identify transgenic plants from a wild-type plant. So we were going to extract DNA from both the plants, the wild-type and the transgenic plant, and then we're going to amplify it using a PCR, and then using the product to identify which one is which using the gel electrophoresis. So the gene of interest is called CAB. It's a chlorophyll AB binding protein, and it is light harvesting complex. It does help with converting the light energy to chemical energy, and it is regulated by the presence of the light. So which means this gene is going to be expressed when there's a light is available. And this is how the plant sense the environment to benefit, to use the light. And you don't need to express this gene if you don't need it. So when there's no light, there's no expression of this gene. To study this CAB protein, we need the reporter gene, and we are going to use GUSA, G-U-S-A. It codes for an enzyme. When it cleaves the substrate, it produces a blue precipitate, and we see it as a blue flower. So today our genotyping experiment is to have 2 lines, 2 plants. One is the wild and one is the transgenic plant. And we need to guess which one has the GUSA transgene. And we used pipetters. They're like devices that we can dispense small amount of liquid. And we use 2 types. One is P20, which range between 2 to 20 microliters. And the other one is P200. The range is 20 to 200 microliter. And we worked on them, we practiced on them before we used them in the real experiment. So the first part of the experiment was to wash the FTA cards. These cards have the leaf smashed on it, and they have the protein and the DNA. It's released from the leaves, and they bind to the card. So we did wash it a couple of times, purify it with the purification reagent, and clean it a couple of times with the buffer. And you can see here the technique with using the pipettes. They discard the tips every time they put a new solution in the small test tube. So we're trying here to remove all the liquid and leave the FTA paper inside the tube as much as we can. We repeated the buffer and the cleaning several times. Sometimes we were making mistakes by adding the air instead of getting rid of it. But it was a very good learning opportunity to use the pipettes. And from there, we added the mix, the PCR mix that has all the primers and the polymerase tag. And we needed that to be on the ice. And now the mixture is ready to go to the PCR. And then the second part of the experiment, we needed to start labeling the beakers before we do any steps. And you can see the student following the procedure step by step. Then collect 3 to 5 seedlings for each of the treatment, and you immerse them in sodium solution and using tweezers, and then you place a screen on top of them, and your group leader will vacuum them so that they remove all the intracellular spaces, and then the solution can go in and the plant will sink because it's like full of soaked with the solution. And then we transfer them to an incubator, And after about 20 minutes, we take out the X-Gluc solution and we add ethanol. And then we put them on a hot plate until the leaves are white. This way we remove the chlorophyll, and it's easier now to observe the blue gas precipitate. And we're going to use the dissecting microscope, and we record the observations and the photos for the seedlings.
So this is wild type, is it?
Yep.
Okay. So here are the little leaves. You see those things sticking out like a antenna?
Yep.
Those are trichomes on leaves. So leaves have single cell hairs on the top. So you can imagine an aphid might not like walking on this. Okay, so I'll take a.
And now for the last part, we need to run the gel for the PCR that's ready. It's been an hour and a half. The PCR amplified our DNA. So we have the gels with the wells ready. And you can see here your friend trying to put the sample and load it inside the well. It is not an easy one. It needed practice. Your hands are going to tremble, and sometimes the gel is going to move, which is okay. We can put back the gel, but the most important part is that the sample goes inside the well, not outside. And you can see here she's moving the gel. And by practice, your hand becomes steady. This is my hand. You can see done. You put the sample and you take it out. It's not that hard after you practice a couple of times. And then we ran the gel. We hooked it up with electricity. The DNA is negative. It's going towards the positive. The gel ran for about 30 minutes or so. And then we took the gel to visualize the DNA bands. We put it into the machine that will read, or it's a fluorescent that will show us all the bands, and we can estimate where is the each of these bands and where's the gene. You can see here there's a positive. This is the ladder, and you can compare it to the ladder and you can see the gene. And this last slide is the 3 different plants with the gene expressed. You can see the blue one, highly expressed GUSA gene. And this is our trip. It was fun. I think we solidified our learning for the PCR and the gel electrophoresis. I hope that you enjoyed it.